Good afternoon, everyone. We have seen COVID-19 sweep across the world, causing unprecedented sickness, hospitalizations, and deaths. Even as we speak, there are communities in many parts of the globe struggling with the disease. Given the gravity of the current situation, I'm honored that you have joined us today for a discussion on the unmet potential for repurposing existing therapeutics in the progression of early COVID 19 infections to severe disease and death. This issue has taken on new urgency given the limited availability of vaccines in various countries and the emergence of certain viral variants that demonstrate resistance to some vaccines. To combat and study COVID 19 affecting communities closer to home, the USC COVID 19 Pandemic Research Center was launched in 2020 with a mission to promote research that allows us to better understand and mitigate the impact of COVID 19 on the diverse communities of Los Angeles, especially those experiencing health inequalities. It is my pleasure to introduce one of the center's co-director, Dr. Howard Hugh. Dr. Hugh will serve as the host and moderator of this afternoon's event. Dr. Hugh is the Flora L. Thornton Chair and Professor of the Department of Preventive Medicine here at the Keck School of Medicine of USC. He is an author on over 300 peer-reviewed publications and has served on many boards, including the Board of Directors for the Physicians for Human Rights, the Board of Population and Public Health Practices of the Institute of Medicine of the National Academy of Sciences, and has chaired the Research Commission for the International Physicians for the Prevention of Nuclear War. Please join me in welcoming Dr. Howard Hugh. Howard?
Welcome, and thank you for joining us today. Last summer, as the pandemic was truly raging, we launched the COVID-19 Pandemic Research Center at USC. Its mission is to promote translational research using multidisciplinary approaches to understand and mitigate the impact of COVID-19 on the health of populations. And as Dean Rao said, our particular focus is leveraging its many collaborations and existing bases of research to understand the unequal impacts of the pandemic on the diverse populations of LA and beyond. The goal is to understand why certain populations have an elevated risk of contracting the virus, why they're more likely to suffer from severe physical and emotional consequences and to die from the disease, and to use the insights generated to reduce and prevent such inequities. Today, we'll be addressing this role of repurposing existing therapeutics to understand how we might prevent the progression of early COVID-19 to severe disease and death as part of the global strategy to reduce the pandemic's impacts. This issue is part of the discussion of inequities and has taken on that new urgency that Dean Rao mentioned, given the uncertainty regarding the effectiveness and timing of the vaccination race, as well as the particularly acute impact on minorities and the limited availability of the vaccines, especially in low and middle income countries. Although the experience with hydroxychloroquine dampened enthusiasm for using existing therapeutics to treat COVID-19, other existing therapeutics have emerged for which promising data suggests they may be effective in preventing the progression of COVID infection, thereby potentially saving countless lives. Today's event is not only the first major public event showcasing the research center we've created, it also is the first grand rounds in population and public health, a new series of high-profile events to be hosted by the Department of Preventive Medicine at the Keck School of Medicine at USC. So without further ado, let's begin our program. Today, we're particularly delighted to welcome Dr. Janet Woodcock, the Acting Commissioner of the United States Food and Drug Administration, who will share her thoughts on the emergency use authorization process for repurposed drugs. But before we hear from Dr. Woodcock, I have asked our very own Dr. Jeffrey Klausner to start off by presenting an overview of the drug fluvoxamine as a case study of a low-cost existing therapeutic that recent data suggests can lower COVID morbidity and mortality. Dr. Klausner is clinical professor in the Department of Preventive Medicine at the Keck School of Medicine here and an expert in infectious disease and epidemiology. He's also a former medical officer for the Center for Disease Control and Prevention and a former clinical professor at UCLA. Klausner and his team have conducted NIH-funded studies and published over 400 papers on prevention strategies against the spread of HIV/AIDS and sexually transmitted infections around the world, and he's a frequent advisor to the CDC and the World Health Organization. Ladies and gentlemen, please welcome Dr. Jeffrey Klausner.
Thank you very much, Professor Hu, for that kind introduction. So I'm going to give about a 10-minute talk about repurposing medications for new treatments, a little bit of a personal perspective, and then highlight some of the more exciting findings regarding fluvoxamine, and then also give an update of where we are with a variety of repurposed treatments that people have been studying. Next slide, please. So based on clinical observation, physicians have a long history of using medications to treat a variety of conditions. And actually, as an infectious disease specialist and researcher, I've been involved with all the different medications that are listed on this slide. And I just want to kind of quickly go through them and give a brief understanding about how these, were potentially developed for alternative purposes. So actually, one of the most notorious medications, thalidomide, which was technically never approved by the FDA, what was found to be useful in the treatment of leprosy in the late 1960s. And because of understanding the mechanism of thalidomide, that it inhibited inflammation, people, including myself, started to study that in tuberculosis and HIV. And ultimately, the drug became approved and is one of the first-line treatments for multiple myeloma. Secondly, a drug called cyclosporin, which is a very powerful immunosuppressant or immunomodulating medication. Before the development of antiretroviral combination therapy for HIV or AIDS, was being studied by myself and others as an immune modulator to reduce the rapid progression from HIV to the AIDS syndrome. A little bit more recently, we've been looking at antibiotics, which have been studied for other infections, such as azithromycin or cefixime, which are inexpensive, generic, readily available antibiotics that are on the WHO essential medication list as an alternative treatment for syphilis. And particularly as syphilis increases globally around the world, there's a desperate need for new treatments. And then lastly, doxycycline, which was actually a derivative of tetracycline, which has been available since the early mid-1950s, and then doxycycline in the 1970s. We've been studying it as a repurposed medication for the prevention of both chlamydia and syphilis. All right. Next slide. So there are actually multiple barriers, which I think we'll hear a little bit more about from Dr. Woodcock and the FDA. There are multiple barriers to the use of generic or old, drugs in terms of the evaluation. Anytime you evaluate a drug, whether it be a clinical trial or look at existing data review, there are costs to that. And so there's high cost to these studies, and there's actually a low return on investment because many of these may be generic and inexpensively priced. So sponsors or manufacturers are unlikely, able to reap the profits that they expect from a new patented medication. There are multiple regulations. So while an old or generic medication it may have been available for 20, 30 years, if researchers want to use it for new indications or in a new type of population, they have to often go to the FDA for permission to do that. And there may be regulations regarding research as well. There's a limited research infrastructure. So one thing we've learned about COVID-19 is the United States was not prepared at all to be able to quickly evaluate new therapeutics. We just do not have an existing research infrastructure that we can quickly mobilize to do new studies. There's the overwhelming bias by physicians, by manufacturers, by the general public that newer is better. And, why would a 30-year-old medication be useful in a new context? And I think also importantly, perhaps until now, there's been a general lack of leadership from major organizations in the United States to really study older medications or look at new opportunities for generic medications. Next slide. So I wanted to bring up a medication called fluvoxamine. Fluvoxamine was first FDA approved in 1994. So, over 25 years ago, it was approved for obsessive compulsive disorder. It has been used and used around the world to treat depression, anxiety, and other mood disorders. It's considered a psychiatric medicine. One major mechanism of action, it's called a selective serotonin reuptake inhibitor, or an SSRI. So similar to medications like Prozac that people may be familiar with. But also importantly, in the past decade, another mechanism of action has been identified as a powerful anti-inflammatory. So as we've done more basic science and understand how inflammation works, scientists have identified that fluvoxamine actually activates a certain receptor inside the cell that is involved with the inflammation process. And by activating that receptor, you actually have a tremendous decrease in inflammation. And people may know that one of the consequences of SARS-CoV-2 infection is an inflammatory response that causes leakage of fluid into the lungs, that causes people to not be able to breathe normally, obviously causes the fever and some of the other symptoms. And if we could change and modulate that inflammatory response, people can do a lot better who get sick after infection. Next slide. So because of those potential mechanisms of action, this drug has been studied, and there was the first report was published in November of 2020 by a psychiatrist group out of Washington University in St. Louis, Eric Lenz. And that's this report on the left, fluvoxamine versus placebo and clinical deterioration in outpatients with symptomatic COVID-19. And Eric's study was quite remarkable. It was, a small to moderate size randomized controlled trial, but in the study, there were 0 out of 80 people who deteriorated in the hospital or got worse who got fluvoxamine versus 6 of 72 who got a placebo. So that essentially was 100% reduction in the risk of deterioration in the patients who got fluvoxamine versus who got placebo. It was a small study, but it was published in a major journal, and it received a lot of attention by physicians and other infectious disease experts. A second study, more recently published this spring, on your right, was a prospective cohort of fluvoxamine for the early treatment of coronavirus disease 19. And in this study, which was an observational study, means it was not a randomized clinical trial, but one of the clinicians from the Golden Gate Fields racetrack outside of Berkeley was dealing with an outbreak of COVID-19 among the trainers and the workers at the racetrack. And he had read Lenz's early study, and he decided as a clinician, which he's allowed to do because the FDA does not regulate medical practice, he was going to offer fluvoxamine to his patients at the racetrack. And 65 people opted to accept fluvoxamine therapy, and 0 of those 65 were hospitalized, versus 48 people were sick and did not agree to go on fluvoxamine therapy, and 6 of those 48 were hospitalized. So this was really exciting to have 2 studies in a row showing that people got the medicine, 0 got sick, versus 6 of 72 or 6 of 48. Next slide. So this actually caused me to think about, okay, well, we have this, major problem of COVID-19, in the United States, early, parts of December and January, we didn't really have a big vaccine rollout yet. Certainly this was going to impact the rest of the world. If we have existing drugs that could help treat COVID-19, how do clinicians know when to use them? So I published this in the Washington Post on January 27th, and really it was kind of supposed to lead to a discussion, from a clinical perspective. When you as a physician, your primary responsibility is for the health and welfare of that individual patient in front of you. You are obligated, first of all, to do no harm, but also obligated to treat that patient and give that patient the best options available. And at the time, there were not a lot of options available for, patients out of the hospital. So to me, it potentially made sense if you had something that was promising and not harmful, why not give it a try? But then there's the balance there between, okay, well, can you make these recommendations for all clinicians and physicians in the United States? And generally, the bar for broad recommendations in terms of changing treatment practices across the population of treaters takes a higher level of evidence. People want to see multiple clinical trials. They want to see large clinical trials and much stronger evidence. So to help me think through this, actually in January 22, I convened a group of NIH and CDC experts and other infectious disease specialists from different institutions around the United States. And overall, when we presented those 2 studies and we looked at the data and the mechanism of action, among 20 individuals that we surveyed who were part of this group, 11 actually supported the idea that, well, yeah, the data are promising. We should be engaging in shared decision-making with our patients. And so we should present the information to our patients in a non-judgmental way, alert them to the risks and benefits, and let them decide what is best for them. And only 5 of the group really opposed that process, and 4 were indifferent, neither opposed or supported. Next slide. But it's important to remember, that, no prescription is completely harmless. That, from something as trivial as people, choking on a pill or potentially, aspirating that pill into the lungs, to some drug-drug interactions that people may not be aware of, to, side effects that could occur. There are always potential complications, and particularly when you amplify that across a large population. So this actually was an editorial that came out last week by Vivekananda Yah, who's the executive director of the Georgia Institute for Global Health in India, where in India right now, a lot of prescribers are treating people with, a bucket load of different medications, from vitamins to antibiotics to anti-inflammatories, without necessarily a lot of strong evidence of the benefit of those in the population. So there is this balance that we always must, take under consideration. Next slide. So right now, this is just a select list. This is not all the medications, but all these medications people may have heard about: hydroxychloroquine, dexamethasone, which is a very powerful steroid, ivermectin, which is historically been used as an antiparasitic agent, zinc, which people often think about with colds and, people have flu-like symptoms, and then fluvoxamine. So right now, the US NIH, based on the available evidence that they've reviewed, does not recommend hydroxychloroquine, does recommend dexamethasone, says there's insufficient data to recommend or not recommend ivermectin. Zinc is not recommended. Fluvoxamine, at this point, they say there's insufficient data. And then an additional drug just this week, colchicine, which is another anti-inflammatory. The NIH group came out and said not recommended. Next slide. So, part of the challenge of this kind of work is who is going to fund it. So fortunately, actually, one of our panelists today took it upon himself to fund in this area and created the COVID Early Treatment Fund, CETF. And this has been a critical group that has supported research on these generic medications. So as expected, you might not expect big pharma to be investing in research, and academic institutions themselves do not usually have internal funds. So it really takes nonprofits and charitable organizations and people with a vision who see an opportunity here to make a difference. And we're going to hear from Steve Kirsch, the founder of the COVID Early Treatment Fund, a little bit later today. Next slide. So I think the way forward is that we need to think about a new regulatory framework for studying FDA-approved safe medications. We're optimistic with Dr. Janet Woodcock at the helm there now that we may be seeing this or reinforcing existing frameworks that she already started as the center director for drugs and evaluation. There are some statistical considerations that we need to think through a little bit further in terms of how do we define what works. Should the bar be a little bit different because these drugs are already proven to be safe by the FDA? And then lastly, we need to make sure that we advocate and we support opportunity for a dedicated government funding mechanism to study the repurposing, because as I mentioned, this is not likely to come from the for-profit pharmaceutical industry. Next slide. All right, well, I think that's my presentation. So I want to thank you all very much. And now we'll move back to Professor Hu.
Thank you, Jeffrey, for that great presentation and provocative presentation. This great segue to now introduce our next speaker, Dr. Janet Woodcock. Dr. Woodcock is the current acting commissioner of the US FDA and director for drug evaluation and research. She's held a number of key leadership positions since she joined the FDA in 1986. For over a decade, she served as director of the Center for Drug Evaluation and Research, which oversees generic drug approvals. She worked as part of the Operation Warp Speed to oversee the development of COVID-19 therapies, including monoclonal antibody treatments for COVID-19 patients. She oversees the full breadth of the FDA portfolio and execution of the Federal Food, Drug, and Cosmetic Act and other applicable laws. This includes assuring the safety, effectiveness, and security of drugs, vaccines, and other biological products for human use. What a pleasure it is to have Dr. Woodcock with us today. I turn the microphone over to you.
Thank you so much. And yes, if I could have the first slide up. So I was asked to talk about repurposing drugs in the time of COVID. What are the issues? I think it's good that I'm following Dr. Klausner because we're going to be singing some of the same song in some different varieties. Next slide, please. So it isn't like interest in repurposing is new, as you have heard. The National Center for Translational Research and CATS at NIH has had a major initiative in this that FDA's been collaborating on. And the advantages of repurposing includes a higher success rate, actually, than using a totally novel compound. There's a known safety profile, wide availability, often not expensive. But the barriers include this frequent lack of intellectual property protection has already been mentioned. So there might be multiple generics or versions available. There's no incentive for the private sector usually to develop these new uses. The Cure ID program or Cure Infectious Disease program, which is a collaborative FDA and CATS program, maintains a platform to capture novel uses of existing drugs. So this is a real world evidence platform where we capture, particularly for tropical infectious diseases, what is successful, because so many of these diseases have no treatment and people are just trying different things. It's accessible by mobile devices. It's publicly available as far as its results. And it's been expanded to capture treatments repurposed for COVID-19. And what are the experiences there? Next slide. So the cost, though, as you already heard, of clinical evaluation is probably the major barrier to getting repurposed drugs out there and widely used. Once a promising agent, say, is identified, as you heard from these small series or small trials, they require evaluation in rigorous clinical trials to be accepted for wide use and recommended by guideline committees or approved by the FDA. And given the current clinical trial ecosystem and the way it functions, this is likely to be a very time-consuming and costly effort. Even if 1 of 10 candidates is successful, the cost may be prohibitive for nonprofits or other interested parties. It's still too costly, usually. So many of these efforts, people try sporadic efforts, but they never reach a conclusion due to multiple logistical, financial, regulatory, and cultural barriers. And I'm going to talk next about COVID-19 repurposing agents, because that's the topic of this seminar, and it's also a good case in point. But I also wanted to mention I was the person under great duress, opposition. I proved thalidomide for E-nodosum leprosum many years ago. And of course, then it became, it spurred a new kind of program in multiple myeloma, among other things. So it shows these repurposed drugs may really have use if you're able to get them studied. Next slide. So what about COVID-19? Well, I think when you think about repurposing during a pandemic or another emergency, what would you really like to do? Okay, I think you'd like to have a centralized scientific assessment of candidates and prioritization for clinical evaluation, perhaps globally. What we saw was all these people were doing in vitro screening of different kinds. They were doing all kinds of other laboratory evaluations, but it might be good to not duplicate those around the world, but maybe have a couple labs study, assign compounds out and conduct unbiased assessments of the screening results to prioritize and nominate candidates. There's strong mechanistic rationale. So many people think for fluvoxamine, there is a strong mechanistic rationale, and so you might prioritize that quite high. And then ideally, have running a large scale, pragmatic, meaning not a lot of bells and whistles. You don't have to do intensive evaluation. Master protocols, you can evaluate multiple candidate interventions rapidly. Try to include as much of the population as possible in these, rather than limit it to some major medical centers or small, couple site type of trials. And the real key of this is, if you have a pandemic, if you could try to randomize the first patient or as soon as possible, start randomizing people so that you can rapidly generate information. And for example, that's what Recovery did for hospitalized patients in the UK. And they learned a tremendous amount of information by doing that. And they ran a very pragmatic trial. And possibly use a stage design. If you have a tremendous number of candidates, as we have had with COVID, can you do screening, a seamless phase 2-3 type of study where you screen them and you have some kind of threshold. And if they pass that threshold, you graduate them into a full efficacy evaluation rapidly without stopping. But what actually happened in COVID? Next one, please. That didn't happen. So prior to development of virus-specific therapies, such as small molecule antivirals that are still being worked on, or neutralizing antibodies, which are currently available under EUA, repurposed agents were the only game in town for treating COVID. There were numerous in vitro screening programs and observational series, and these led to hundreds of possible candidates. And of course, people were using erythromycin, they were using this, that, hydroxychloroquine, this, that, and so we got all these series, got people who were enthusiasts about various treatments, and then they did other small series. Some drugs were used extensively for treatment well before we really knew whether they were going to help or not. Hundreds of trials are set up worldwide in order to evaluate candidates. And I'm not sure how candidates were prioritized for these trials. We did have a rigorous prioritization in Operation Warp Speed and for the active trials that were done at NIH, but I'm not sure what methods were done elsewhere. Next one. So early in the pandemic, the Center for Drugs that I was heading at the time commissioned an inventory of trials, which we kept up. And we identified more than 2,000 trials with about 2,800, 2,900 treatment arms worldwide that were going on for therapies. The proposed enrollment of these trials was over 500,000 people. Represents about a third of all trials that were done for COVID within the total, which would include like observational trials, vaccines, and so forth. And this increased over time, and there were, the therapeutic classes that were studied shifted over time. Next slide. I'll try to get through this quickly so we can have some discussion. But you see that, for example, antivirals were not very broadly used, although there were more than 100 trials for hydroxychloroquine around the world. But you see they were immunomodulators were used broadly, and these are all primarily repurposed agents until the monoclonal antibodies kicked in. Next slide, please. So the problem is that regulatory actions and clinical guidelines, as you already heard, the recommendations, say, of the NIH Guideline Committee are evidence-based, and they want robust evidence before making wide recommendations for population. And so we looked at all these trials, these almost 2,900 trial arms, to see whether they were randomized and sufficiently powered to be useful for regulatory decision-making or for guideline use. And so because you can't tell from the registries around the world whether the trial is sufficiently powered, we looked at the planned enrollment and made some assumptions about adequate power. Next slide. And what we did a sensitivity analysis because this was pretty arbitrary. We decided you needed X number of patients for this situation, right? And so we did a sensitivity analysis, which may be kind of hard to see here. But the bottom line of this is that even if you reduce the number quite a bit for power, required for power from what we had proposed, it's still, most of the trials did not meet that threshold. Next slide. So this is not very, very clear. Can you clear up the slide a little bit, possibly? Basically, this shows that master protocols were generally adequately powered, and most of the other trials outside of the master protocols had rather small enrollment. So although the master protocols were only a tiny bit, the bar on the top of the total trials, they took a large percentage of the population. But the bottom line here is what we found, that of all the trials that we looked at, only 5% of them could be considered both randomized and adequately powered for the stage of disease they were studying. So these were repurposed agents. And I think this is why we see all this controversy to up till now about which agents are useful and effective in different stages of the disease, because many of these trials have not been adequately powered. Not only that, most of them didn't enroll as many people as they intended to enroll, possibly because there was so much competition for patients at these medical centers. And so even though they may have been marginally powered, they ended up not even enrolling the number of people they aspired to enroll. Next slide. Good, that's much clearer. Next slide, please. So bottom line, repurposing efforts during the pandemic were far from the ideal state. Only 5%, as I said, could really likely yield actionable results. And that was globally, but also in the US. There was no difference really in the rate of actionable trials. And of these 5%, as I said, many failed to reach their enrollment target. As usual, most patients in most communities in the United States did not have access to a clinical trial, and this decreased diversity and representativeness. And here we had tens of thousands of people being hospitalized and actually many being dying, and we had trouble enrolling the trials that were set up. And this shows that there's really some problems with the clinical trial ecosystem in the United States, and gets back to Dr. Klausner's issue of we really don't have the capacity to evaluate repurposed drugs very well. So, and with the resources we had, many of them were squandered on non-informative trials, and this slowed evidence generation. The majority of conclusive evidence comes from the platform trials and master protocols that have been done around the world. Next one. So as I said, there were over 100 trials of hydroxychloroquine initiated in the early stages. Many of them were inadequately powered and didn't reach target enrollment. Same with convalescent plasma, same with many of the other agents. Duplication primarily occurred with repurposed drugs because they were available and they had a known safety profile, and so people could easily initiate studies. The lack of some sort of coordination or collaboration led to many investigators commencing research on their own. And despite the numerous trials ongoing, as you heard, many candidate repurposed drugs were not able to be studied. And so, although we had all these trials going on, we had really a limited number of repurposed agents that were looked at. Next slide. So here's the link to, we published this result, and this is the link to the article. Next slide, please. So, therapeutic development for the virus really did encounter huge challenges. The healthcare systems were overwhelmed, and so they didn't really have the ability to do a lot of research. There was lack of sort of fit for purpose, ready to go clinical networks. Typically government NIH run studies may take up to 18 months to set up. We did much faster than that with the active trials, but still it took a while to get them going. And frankly, this disease is tricky. And I think we underestimated at the beginning. Each stage of the disease, say the early stage, the pre-hospital worsening, the early hospital stage, the on-the-vent stage, the post-discharge stage, they have very heterogeneous outcomes. This means that you need a lot more statistical power. You need a lot more patients to have adequate statistical power to find a signal in this kind of haystack. Even fairly large trials, as you know, have had conflicting conclusions about what to do. And we had a large number of underpowered trials that hampered enrollment in the medical centers. Next slide, please. Again, we're having a little trouble with the slides here. Next slide. They're getting, could you go back one and try to get it in focus? Okay, I think it's probably very hard to read this. So why don't we skip over to the next slide?
Thanks, Janet. We're almost done.
There you go. There you go. All righty. So other things that we discovered during this pandemic is that the fastest accrual came from industry-sponsored or CRO-supported sites. And I want to quote Martin Landry, who's one of the PIs of the recovery trial, with his permission. He said the rate of enrollment in a trial site in his study was inversely proportional to the number of professors. Most medical centers now are in a web of affiliated hospitals and an integrated health system, but most of those community hospitals are not engaged in clinical research, and therefore they lack the training and study personnel to be able to contribute. So we had all these patients in the community hospitals, and we couldn't recruit them into the community, into the research. And the case studies, and open label trials yielded incorrect information. And as was already said, the US care system has a treatment bias and so forth. Next one. So I'll try to wrap up quickly. I'm sorry. Yeah. The master protocols, as I said, perform very well, but there are difficulties about, getting these going in an emergency. They're hard to set up at the beginning. Next slide. So I think what we really need to do is, as I said, develop a robust screening mechanism and then have a system that can generate definitive, highly actionable information. Next slide. We need a broader community clinical trial network, one that can respond rapidly to outbreaks and other emergencies, that can enroll people where they live and obtain healthcare through the providers they trust, enroll diverse populations, especially those disproportionately impacted. The same issue occurs with many chronic diseases. This isn't something that needs to be stood up in a pandemic. This is something that we need all the time. And they could run master protocols that looking at chronic diseases and the best treatment protocols for those. Next slide, and I'll finish, sorry. So this really applies to repurposing drugs as well. What we saw in the pandemic is those such as REMAP-CAP, which it was a community acquired pneumonia network, were able to rapidly reconfigure themselves and start studying repurposed drugs for the hospitalized phase of COVID. If we had master protocols set up and running in the community, we would really be able to much more quickly evaluate out of the huge armamentarium what would really work for any given outbreak. And I think that my last slide. Next slide. So drug repurposing is a key endeavor because right now most drugs are probably used suboptimally, right? But there are many barriers. The SARS-CoV-2 pandemic illustrated the weakness of the global clinical trial ecosystem. We were not able to rapidly evaluate repurposed agents, particularly for outpatients, and the multiple fragmented efforts yielded few actionable results. And we need to take some lessons learned from this. Thank you very much.
Thank you very much, Janet. This is a time we're going to pivot directly to our three panelists who can provide a response, perhaps ask questions. We're going to start off with Dr. Neha Nanda, who heads the Healthcare Epidemiology Unit. She's an amazing clinician, an infectious disease attending physician. Steve Kirsch is an entrepreneur and founder of the COVID-19 Early Treatment Fund that Dr. Klausner referred to. And Dr. Neeraj Sood is a professor and vice dean for research and faculty affairs here at the Price School for Public Policy and a founding member of the USC Schaefer Center. He does research on economic epidemiology, infectious disease, and is currently leading studies on COVID-19 in collaboration with our department. So let's start off with Dr. Neha Nanda with some remarks.
Thank you, Dr. Hugh. Thank you, Dr. Woodcock. That was really very helpful to put everything in perspective. I would love to hear your thoughts in comparing the trajectory that monoclonal antibodies took relative to, let's say, the drug that Dr. Klausner talked about, fluvoxamine. There are some things that are so obvious, and we know how we can do better moving forward. But I just love to hear what you have to say, how we can do better next time around.
Right. Well, I was the therapeutic lead for Operation Warp Speed, so I think it is quite reasonable to ask me that question, right? The monoclonal antibodies were being developed and needed to be evaluated. They're highly effective when used early in disease, but of course, they're not logistically very useful. And the holy grail here is to get an oral agent that can stop this infection in its tracks and prevent progression to more serious disease. And so right now, Active is setting up an outpatient trial that is kind of a hands-off trial, looking at more repurposed agents, a pragmatic lower touch trial. That would have been desirable to put something like that together earlier, I think, so that we could move many agents through. But as was already stated by Dr. Klausner, I think this was, there were so many agents and it was very difficult to figure out the prioritization. And it isn't sort of the culture to do it that way, but that is a lesson learned. We should have put together a highly pragmatic trial to probably phase 2-3 seamless, okay, a screening trial followed seamlessly by those that passed into a pragmatic trial. But I will say, I think all you ID docs, right, they want to do all these antiviral tests and all these viral, PCR tests and all this kind of stuff. And pretty soon you have some that only can be done in a medical center. And we had a terrible time even getting the, PCR equipment and reagents available and all this stuff. And the more science you add to this and the less pragmatic you are, the fewer agents you're going to be able to evaluate. So in some cases, we have to take off our really sciencey hat and think pragmatically, how can we just evaluate these and see if we can find some agents that reduce progression.
Can I make a comment? Because I think what you ended with being pragmatic it would have been so helpful. Let's look at the time. In November, December, we had patients who did not qualify the criteria for monoclonal antibodies, right, because we have, a set criteria based on, right, Blaze trial and a couple of other trials.
Right.
And there was a need for something like this then.
That's right.
Now today, with the positivity rate of, in LA County, 0.4%, we are not even using our monoclonal antibodies. And as you know, now the pendulum has swung towards let's find therapeutics around post-COVID, if there
right.
are any.
Right.
But you're right. I mean, it's all about getting pragmatic and answering to the need of the hour.
Right.
Let's hear from Steve Kirsch now. Steve? You're on mute.
Yeah, sorry. They won't let me start my video.
All right.
Okay, so now I can, here I am. Hello. Hi, Janet.
There you are.
So, yeah, Jeff introduced me earlier. I started the COVID-19 Early Treatment Fund, and we sponsored all the fluvoxamine trials. And so I'm an expert. I feel I've got my expertise in School of Hard Knocks in terms of running trials and enrolling people and how difficult that is. And I have a lot of war stories I could tell, but that would take the end of the seminar to do that. I wanted to start though by really thanking Janet. I've got to know Janet over the months since I started CETF, and I can tell you that for all the people in the United States government that I know, I would rate her at the top of the list. She's extremely responsive. She's very knowledgeable. She's got to be an expert in all these categories. And I can't say enough about her. I asked Jeff if I could talk for 3 minutes about and use all my time thanking her, but he said, no, I only have a minute. So anyway, thank you, Janet. You're absolutely phenomenal. I've really enjoyed my chance to interact with you. And I would welcome any opportunity to meet with the FDA to talk about ways that the system could be made a lot better, because I'm on the other side of this, and so I see a lot of the problems. So I think I could help out in that. Like, one observation I had is that if you have 0 professors involved, then you'll have infinitely fast enrollment. So that, I think we get a lot of drugs through that way. So I wanted to just talk about a couple of things. One is there's something called the precautionary principle in medicine, and using all the available evidence. And we use these principles sort of differently. For mask wearing, we use these principles correctly, in that we say, well, mask wearing can't hurt. We don't have enough evidence to support it, but we're going to do it because it may provide a benefit that doesn't hurt. But when we deal with the repurposed drugs, we actually take the opposite approach. Instead of using all of the available evidence, we're just using the very highest tier of evidence. And instead of saying there's no harm, we're kind of doing the reverse and saying, let's not use this until we have the highest level of evidence. So I think there's an inconsistency there that I think if we made it consistent and we did the same thing for mask wearing as we did for repurposed drugs, we would be in much better shape today. And as an example, there's a practice, Fareed and Tyson, who've treated 6,500 patients of COVID. And if they get the patients within 24 hours of first symptoms, nobody goes to the hospital, 0. And so, but nobody believes them, and nobody goes out to find out. So one suggestion is, don't just look at the data coming in, and don't just look at the published studies and peer-reviewed journals, but go out, and when you see some anecdotal evidence of 6,500 patients and 0 hospitalizations, find out what they did and take action on that. So I think if we did that, we could dramatically lower the death rate, and that still applies even today. And finally, just in closing, I wrote an op-ed on Trial Site News called "Should You Get Vaccinated?" And it talks about a lot of these issues, the pros and the cons. And I'd encourage people to look for that if they're interested in how we can do better. It explores a lot of the issues with respect to vaccination versus early treatment and the combination.
Thank you, Steve. Janet, you want to give a brief response before we turn to Professor Sood?
No, I'm fine. That's fine. Thanks.
Okay. Thanks for those remarks, Stephen. Neeraj.
Thank you, Harvard, and thank you, Dr. Woodcock. So I think, like, the big picture, the problem is that we don't have the incentives to invest in large clinical trials to repurpose drugs.
Right.
But there are some ideas out there, right? So for example, you could do priority vouchers, transferable priority vouchers, or transferable exclusivity extensions. So if you discover a new use for a drug, say for COVID, you can get a 1-year extension on a cancer drug you have on the market, and that 1-year extension might be worth, a lot of money. And so if you now create those incentives, there's enough expertise in the private industry to now figure out which are good candidates, conduct the large clinical trials, and produce the evidence that a regulatory agency like FDA would need. So my question is, is the FDA advocating for such approaches, or is it open to such approaches or not? And my second question is kind of related to an issue that Steve brought up, that you said, we have a treatment bias. We have clinicians who are willing to experiment or try out a lot of different treatments. Well, that generates a lot of real world evidence. Are we mining Medicare claims data, or, electronic health records, or other data sources to see what works and what doesn't work? It's not a clinical trial, but, we know clinical trials also have limitations. So this is in some sense more pragmatic, real world. We can use fancy statistics to reduce bias, but maybe that's an approach we should kind of look at, especially where there are no therapeutics and the disease burden is high, where, like we need a different approach. Our risk benefit trade-off might change there.
Well, on the first one, the trade-off, you're talking about trade-offs, okay? So you give extension of exclusivity, you give priority vouchers, you're trading certain things off. It's really up to Congress. That's a societal type, and everybody wants a priority review voucher for their disease, for their development program and so forth, for rare diseases, for this and that. So eventually these incentives don't become that meaningful. When there's so many of them available, they aren't worth as much. But FDA cannot create those. They have to be created by Congress. And I think they would ask our opinion. However, that's the administration's opinion, really not the FDA's opinion. Now, on the real world evidence, FDA has a large effort going on called the COVID Accelerator. And that's with the Reagan-Udall Foundation, working with a lot of the data aggregators to see if they can learn from all this real world data. My problem with this, okay, is that we've had moderately sized trials of many agents that have been done, placebo-controlled trials, and we still can't figure out whether the drug works. And those folks who are ID docs here and so forth know this, okay? That's because the disease has so many disparate outcomes and it's so variable and so forth. And so in that situation, unless you have a drug that's a home run, which some folks think fluvoxamine may be, okay, unless you have a drug that's a home run, you're not going to detect that treatment effect in the middle of so much noise in a real world evidence, no matter how much fancy propensity matching you do, okay? And you're not going to convince anybody, right? Because we've had all these series and all these testimonials and they've been wrong, okay? So this disease is a tough disease. This isn't pneumococcal pneumonia and penicillin, okay? This is, where you really didn't need a placebo group. So that's my opinion. It's just, even the monoclonals, which had in the high risk group, they're about 70 to 90% effective in reducing hospitalization and death, right? And yet you needed 1,000 patients to demonstrate that because the event rate was low. Because most people don't get hospitalized. So, it's really hard to tell, but we are looking at the real world evidence and that effort is ongoing.
I just wanted to chime in there. Sometimes you have a phase 2 trial and it enrolls people in 3 days on average. And then you do a phase 3 trial and it's mail order and you enroll people in 5 days. The difference of a day or two can make a critical difference in terms of the effect size that you have. Like George, he has 100% rate if you come in within 24 hours. And so those sorts of things are really, really hard to do. And they're hard to do at scale to replicate it. And so a lot of times people look at the phase 3 and say, oh, you only got a 30% effect size. See, it proves that your phase 2 was not 100%, but in fact, they were different. And a lot of people gloss over that timing of drug administration and the amount of drug is everything in this disease. And that's what people have found.
Yeah, well, I don't disagree with you. It's a progressive disease and some people are going downhill and their viral load is not being cleared by their body and they're triggering the inflammatory response. And so that timing is everything because they've had the disease before they had symptoms. And then, but some people go downhill very rapidly after they get symptoms and other people might be 10 days or so and then they get ill. So this, it's a tough disease to study.
Folks, it's 12:59. We're going to have to wrap up pretty soon, and I know that Dr. Woodcock's folks are very anxious to move her along to the next event on her agenda. The chat room has been interesting. There's a whole list of all sorts of questions that have come up. Maybe we'll forward some to you, Dr. Woodcock. But before we get cut off ourselves, I just want to thank all of you, especially Dr. Woodcock for her presentation, but all of our panelists and, of course, the audience for participating in this really interesting conversation, which we haven't heard very much in all the major national seminars and other things that have been going on. Would love to get your opinions on ivermectin and ivaptidil and all sorts of other drugs that have come up. And there's even these interesting suggestions of creating a national IRB to accelerate things. But it's a fantastic conversation, and I know you'll do a fantastic job in stewarding us through the next phase of what we need to do to create this clinical ecosystem of trials for the next pandemic, and maybe this one too. So thanks, everybody. It's 1 o'clock.
Thank you.
Mindful of everybody's time. Wonderful to have you all.